Today we're going to be talking about linkage and what that means for ratios that do not follow the expected Mendelian ratios. And this is section 5.3 in your book. Basically what we need to consider is the question, what's going to happen to our Mendel ratios if the genes are located on the same chromosome? If they are, we would call those genes linked. So linkage is referring to genes that are found on the same chromosome. Here's an example of a chromosome and showing us that A and B are two genes located on the same chromosome, and they are linked. But A is also linked to C, D, E. All of these genes would be considered linked genes because they are found on the same chromosome. Because they are on the same chromosome, these genes usually do not separate during meiosis. So they are not going to have to follow the rules of independent assortment or segregation. These genes will be packaged in the same gamete, so they will be inherited together. These genes will not show the expected ratios that Mendel would predict for two different genes. Remember that when two genes are involved, we might expect a ratio of 9:3 to 3:1 or 1:1 to 1:1. But because genes that are linked are on the same chromosome, we will not see these expected ratios. This idea was discovered in the 1900s with some experiments with pea plants. The original experiment took a purple long pea plant and crossed it with a red round pea plant. Now if you look at the genotypes here, you see that you have heterozygous, heterozygous, crossed by homozygous recessive. This would give you an expected 1:1 to 1:1 ratio. You would expect equal numbers of the four different phenotypic combinations. You would expect equal numbers of purple long, purple short, red round, and red long, because they would all equally recombine. However, this was not the result for this particular trait. Instead, we ended up with a lot of purple longs and a lot of red rounds, about 350 of each of these types. These were, not coincidentally, the same as the parent phenotypes. So purple long and red round are considered the parental phenotypes, and these two were more common. We did have a few of the non-parentals. We did have a few purple rounds and a few red longs, but much, much less. In fact, we only had about 25 of each of these. So we did not see equal numbers. We did not see a 1:1 to 1:1 ratio. Instead, we found a lot of parental phenotypes and very few non-parental phenotypes. The conclusion from this experiment is that these genes did not separate during meiosis like we have seen before. Instead, the genes actually looked like they were traveling together. The purple stayed with the long and the red stayed with the round. The genes then were determined to be located on the same chromosome. We call this linked. Because they traveled together, they must be traveling together on the same chromosome. However, there were a small number that mixed up, a small number of non-parentals. We concluded that this was due to crossing over. Crossing over is when the homologous chromosomes switch small segments, and you can see how this might lead to new combinations. But this is not very common. The results are called recombinants. You can see them there in that picture. And they do account for the numbers of non-parental offspring. However, remember, the numbers of non-parentals are usually much, much, much smaller than the parental types. Thomas Hunt Morgan is a scientist who's very famous for his work with linkage, and he worked with fruit flies, as seen in that picture there. What he did is he looked at the number of chromosomes in a fruit fly. It was a very small number, so it was pretty easy to do some experimentation here. And one thing that he wanted to do was figure out what genes were linked on the fruit fly chromosomes, and then to figure out where they were located on the chromosomes. He decided or discovered that there was a relationship between the crossover frequency, how many non-parentals there were, and how far apart they were on the chromosome. And he used the number of crossovers or number of recombinants to build a linkage map. He basically created a map that was a diagram to show the order of the genes on a chromosome and a relative distance between each. He measured the distance in something called a map unit, and it's sometimes called a centimorgan after him. Basically, what is set as the standard is that 1 centimorgan or 1 map unit is the same as a 1% recombination. Genes that are far apart cross over more often, so their percent of recombination would be higher. Here's an example of the fruit fly chromosome that he mapped, showing different parts of their antenna, different characteristics of wings, legs, eyes, and so on. Basically, we can use his standard to solve some problems involving linkage mapping. For example, if we do a cross where it's homozygous recessive for R and homozygous recessive for E, and we cross it with one that is heterozygous for both, we find that 96% of our offspring resemble one of the parents. So 96% of our offspring are parental. Only 4% are recombinants. We use that recombination frequency to determine how far apart they are. So we would say in this example that R and E genes are located 4 centimorgans apart or 4 map units apart on the chromosome. That's because 4% of the offspring were recombinants or non-parentals. Here's another example showing how you might line these up on a chromosome. If we do either a calculation or a problem tells us that gene X and Y have a 10% recombination rate, X and Z have a 4% recombination rate, and Y and Z have a 6% recombination rate, we can figure out how they are located on the chromosome. Obviously, X and Z are going to be closer together than X and Y. So you're going to draw a chromosome, and we're going to kind of label 3 spots on here. We see that X and Z are only 4 apart, Z and Y are 6 apart, and then when you put that together, you see that X and Y are 10%. They are 10 map units apart. So this is how you would use that data to determine where they are located on a chromosome. We can also do a problem where they give us even more information. Here they're telling us that we have a tall yellow parent crossed with a short green parent, and they give us actual numbers of offspring. So 135 are tall and yellow, just like one of the parents. 140 are short and green, just like the other parent. However, we do have a very small number of recombinants, or non-parentals. 25 are tall and green, and 20 are short and yellow. Right away, we would notice that this is not a 1-to-1 to 1-to-1 ratio, so we know that we're dealing with linked genes. The parental types in this case are tall and yellow, we have a high number of those, and short and green, and we have a high number of those. Our recombinants are going to be short and yellow and tall and green. In order to calculate a recombination frequency, we are going to need to calculate percent of recombination. So we're going to put the number of recombinants, they are circled above, 25 tall green and 20 short yellow. And we need to divide the number of recombinants by the total number. This is what that's going to look like. We're going to take 20 plus 25 and put that on top. Those are the ones that are circled because they are recombinants or non-parental. We are going to divide that by all of the offspring, all 4 of the numbers added together. So that's 135, 140, 25, and 20. When we add those up, we get 45 divided by 320. That gives us a percentage of 14%. So this gene pairing has a 14% recombination rate. That tells us that they are 14 map units apart on the chromosome.